Good morning, and thank you for joining us today. I'm Betsy Collins, the Director of Donor Relations at the Salk Institute for Biological Studies, and I manage the Salk Women in Science program. We're so pleased to partner with the Del Mar Garden Club to bring you an informative event focused on the study of plants, an initiative led by Salk Professor Joanne Chory. Our topic today is fighting climate change with plants. We are grateful to each of you for spending the next hour with us to learn more about the Salk Institute and the Del Mar Garden Club, and hear firsthand from Professor Chory. Before I introduce our guests, I'd like to share a few logistics about today's program. At the bottom of your screen is a bar, and you can see the Q&A box. If you have any questions for Professor Chory or a technical issue, please type in this chat box, and our Salk team will do what they can to resolve any issues. It's now my pleasure to welcome two of our Del Mar Garden Club members who partnered with Salk to bring you today's events. Mara Bickett, Del Mar Garden Club's president. Mara?
Thank you, Betsy. The Del Mar Garden Club is delighted to be partnering with the Salk Women in Science on our annual community outreach program. It's an honor to have Professor Chory share her important research with our local garden clubs, neighbors, and environmental organizations with over 265 registrants joining us today. One of the benefits of the Zoom format over the past year is that our club has been able to go a little bit deeper into education and the science of plants with access to some remarkable speakers. We are proud to have the Del Mar Garden Club's past president and one of our own Women in Science, UCSD researcher Dr. Candace Cole, host the Q&A period after Professor Chory's talk. As Women in History Month winds down, it's exciting to hear about the historical work women like Professor Chory are doing to address some of the urgent problems our planet faces. I would now like to introduce Candace Cole. Candace?
Thank you, Mara. This is a wonderful opportunity to participate in this joint event and to hear Professor Chory speak on her latest research. I would like to give special thanks to my dear friend, Catherine Riviere, Professor Emerita and a founding member of the Women in Science program at Salk, for her help in orchestrating this collaboration, as well as for her tireless dedication to encouraging and supporting women in science. Betsy will now continue with the introduction.
Thank you, Mary and Candace, and all of the members of the Del Mar Garden Club for partnering with Salk Women in Science. Before we introduce Professor Chory, I'd like to provide a little history and information about Salk for those of you who are joining us for the first time. As an organization, Salk has a long history of virology and vaccine research, including the tremendous work by Jonas Salk himself that led to the discovery of the polio vaccine in 1955. In search of a place to build a biological research center, Dr. Salk was encouraged by then San Diego Mayor and polio survivor Charles Dale to come to La Jolla. The city of San Diego donated 27 acres of land, and the March of Dimes contributed $20 million to develop the institute. I'd also like to share with you the many ways to get involved. We offer many programs such as those you see listed on the screen, and serving as an advisory committee, attending an event, or supporting groundbreaking research are great ways to get involved. As I mentioned earlier, I manage the Salk Women in Science program, and our mission is to support the advancement of women in science and technology. Our program provides crucial support for the advancement of women scientists via research grants, travel awards, and child care subsidies. We offer many events to educate the community about the groundbreaking research being conducted at Salk, as well as programs that mentor the next generation of great scientists. In 2018, we established the first Women in Science Ursula Belushi Trailblazer Award, which Joanne Chory was nominated for this past year. However, due to the pandemic, we've been unable to celebrate this achievement with her, so congratulations, Joanne.
Thank you, Betsy.
Without further ado, it's now my distinct pleasure to introduce our speaker for today's presentation, Professor and Director Joanne Chory. Professor Chory joined Salk in 1988, where she is the director of the Plant Biology Laboratory, a Howard Hughes medical investigator, She holds the Howard H. and Miriam R. Newman Chair in Plant Biology, as well as directs the Harnessing Plants Initiative. She is known for her studies that have shown how plants alter their shape and size in response to changes in the environment. Dr. Chory has served on numerous advisory committees and editorial boards and has received multiple awards. Please welcome Professor Joanne Chory. Joanne?
Thank you, Betsy. Let me get set up here. There we go. So just I'll give a plug for the Women in Science program while we're waiting this to launch. It's a great program, and all the young women really love having it as part of the Salk's, philanthropic pool of programs, and so it's been very successful due to all the hard work of Betsy and her team. So thank you, Betsy, for doing that for the few women scientists at Salk. There we go. So where we at? Over here. Okay, here we go. Okay. Excuse me. All right. So today I'm going to tell you about this new initiative we have for the last few years at the Salk, and it involves a plant team of scientists, and we're just a very small group of people. And what we decided to do was actually really get involved in one of the world's biggest problems, and that's climate change. And we had an idea about how we could use plants to actually fight climate change. And so I'm going to tell you about our idea today and how far we've gone with progress on the project. So, as I said here, it takes a global village to solve a global problem. So climate change is a global problem for sure. And right now, we are at a crossroads. Oh, wait a second. Where am I? That's not the slide I want. All right. I skipped a slide.
You want this slide, right?
Yes, I do. Sorry about that.
It's okay.
Okay, so we're at a crossroads, and humanity is at a crossroads. We have a burgeoning population, and this has caused a huge demand on the earth. And so we have increased demand for food, feed, fuel, and fiber. We have effects from global warming, which stressed out our crops that we need to feed people. And we also have a lot of collapsed ecosystems around the planet. And so, as a result of this is all a result of this increased CO2 emissions that we have because we have so many people on the planet, almost 8 billion right now. And so, let me see if I can do this. Getting there. See, I skipped the slide again. Oh, there it is. I'm sorry. Okay. So, what I'm going to tell you is that this is an urgent problem. Our action or inaction in the next 10 years is going to determine our fate, the fate of humanity on the planet. And so, let me tell you how we came up with this idea. So, we're plant geneticists, and you've all heard about CO2. CO2 is a greenhouse gas, and it's kind of like the antagonist in a novel, right? And so it's the bad guy. But when I think about CO2 as a plant geneticist, I think about it as a good thing. It's like fertilizer for the plants. And so let me tell you, so this is because plants take CO2 out of the air, and they suck up water from their root systems, and then they use the energy of sunlight. And when they do this, they make biomass. And so we're a carbon-based planet. All of biomass, all of you and I and everything around us is carbon-based. And all that CO2 that's in the atmosphere is what gives rise to all the biomass on Earth. So the biomass is a problem because we have this global cycle. And we don't have to go over the numbers so much today, but every year, photosynthesis takes up a lot of gigatons of CO2, and then the plants grow, and then they die, and people die, and a lot of the CO2 the plants have taken up gets released back to the atmosphere, and that's part of the problem. And the big part of the problem is us. So here we are, 37 gigatons released by humans each year. And what that's done is set this global cycle, this carbon cycle, out of whack. Okay, so let me show you how out of whack it is. Okay, so here it is over here. In the upper left-hand corner, you can see every year we put up 37 gigatons, but 18 gigatons are in excess of what the planet can actually do something with it. And so this has now caused global warming, and all this was documented by a colleague of ours who lives in Del Mar, David Keeling, in famous Keeling curves. And you can see that curve goes up, and it's been going up since the Industrial Revolution, and it's over, the amount of CO2 in the atmosphere in parts per million is now like 420 or more. And when humans evolved, it was 260, and when the Industrial Revolution started, it was 280. So we're way up above. So not only do we have to not put up more CO2, we have to actually take some out that we've already put up. So that's going to be a big issue for us, and that's what our initiative is concerned with, that aspect of CO2 in the atmosphere already. So here, let me show you what's next. So here's our idea. So we have this idea that the whole problem is a distribution problem, and all we need to do is get plants to put a little more carbon into stable molecules that don't get broken down every year when the growing season is done. And so we have a molecule, and that's called suberin, and it's a natural plant polymer, and it's known to be very stable. And so here is a normal plant on the left-hand side of the screen, and you can see the plant sucking up CO2 from the atmosphere, it's taken in the water and stuff, and then at the end of the growing season, the plant dies, and a lot of the CO2 gets back up in the atmosphere. And so here's our idea. So our idea is if we modify plants just a little, we will have these ideal plants. And these ideal plants will suck up the same amount of CO2, but less CO2 will be released back out to the atmosphere because we've now redistributed into these stable polymers which are in the roots of plants. So we figured in order to change our plants, who are very good athletes, into Olympic kinds of athletes, we have to change a few traits. And so here's the traits we're going to change. So we want the plants to make more extensive roots. And this is because these plants will be more resilient, but they'll also be fixing more carbon into the root. And the root is the part of the plant we're interested in, because we're going to leave that in the soil when the plants are done their growing season. So we're going to do all this with crop plants, I should tell you this. And these are our food plants, and this is because these plants take up almost all the arable land on Earth. And so we're very interested in growing these plants everywhere. And so we want to do it with crops. Okay, so we need to have plants with bigger roots. We need to have plants that have deeper roots. And this is because we want the plants to bury this carbon that we're sticking in this molecule, suberin, deep down in the soil for us so that it will stay there and it will not go back into the atmosphere. Okay, and then the third thing is we need to have what we call recalcitrant biopolymers. So plants make a lot of these biopolymers. You might know lignin. Lignin is what paper is made out of. And that's pretty stable. But this molecule suberin is really stable, okay? And so that's the one we're going to shoot for. And so let me tell you more about suberin now. All right, so. Okay, so we have model plants, and model plants are good because these are the plants that we've used to actually work out all the mechanisms by which plants do what they do. And so this is a model plant that we use, a Arabidopsis thaliana, and we've used this for a long time. It's a tiny little plant with a tiny little seed. It's in the mustard family. It has a small genome, and it has good genetics for looking at segregation of traits. And so you can also see here that it's actually a really good model to study natural variation of root growth. And so these are Arabidopsis plants that we harvested from around the world and brought them to the lab, and you can see how the roots are growing. That these are all in the same time lapse. Roots are growing at way different rates, and some of these make deep roots, some of these make shallow roots. And so, I mean, these are all good material for us to try to get at the genes underlying the traits for more and deeper roots. Okay, so let me see what comes next. Okay, so we've made a lot of progress since we proposed this idea of finding some genes that are in controlling these traits of more and deeper roots and more recalcitrant carbon-rich biopolymers. So here I'm going to show you more and deeper. I don't remember which one I'm showing you. Okay, I'm showing you suberin first. Okay, so what is suberin? You're saying? So suberin is basically cork. So it is cork down in the lower right-hand corner, and you can see it there. And so cork is really recalcitrant to being broken down. Those are all dead cells in cork. And so suberin also grows in the skin of citrus fruits. It's in avocado skin, it's in potato skins, but every plant makes it in their roots. And so it's the root suberin that we decided we were most interested in because we thought we could pack a little more in there, and then the plant wouldn't break it down. And so anyway, so this is why we picked suberin. So this is the structure of suberin biopolymer, okay? And so what you see in the middle, all those little black dots, those are all carbons. So this is like the greatest carbon storage device we could find. And there's very little oxygens, which are shown in red. And so microbes like to come in and decompose plants, add oxygens. So when you see red, you're going to see those things get biodegraded. But in the middle part of this molecule, you'll see there's almost no reds at all. So this part of the biomolecule is very stable, and it's also carbon-rich. There's hundreds of carbons there. And so this is what we want to use to store the extra carbon. So how are we going to get plants to make more suberin? So it turns out suberin, this is all work from other labs that has been done a while ago now. Suberin has two rich layers in the root. Okay, so one way to make more suberin is to make more suberin-rich tissue layers. And we can do that, actually. So we have this one layer called the endodermis, and we now have that as a double layer. We can find genes that make that become more and more prominent. And then the other way you can make more suberin is to get more suberin in each cell that's making it. And so let me show you what that looks like. Okay, so here we see a cross-section through a root. So you're cutting through the root this way, and the root's going down. And you can see where these red cells are, those are cells that contain suberin, because now we're using a dye that's specific for suberin. And so you can see that there are two nice sets of cells in concentric circles that are the suberin-containing cells. So it turns out if we just make more suberin everywhere, the plant will basically kill itself. It starves itself to death. So we can't just do that. We have to make cells that want to have suberin in them to become the suberin-containing cells. And we found some genes that control that trait. And so now we can put those genes into crop plants, which is the plan. And that's going to take us another couple of years. And then we're going to be able to measure it in field trials whether or not these plants are actually storing more carbon. Okay, so that's an example of this one plant, Lotus japonicus. Okay, so how do we get more suberin per cell? So suberin is this big molecule and has a complicated biosynthetic pathway. But you can see from this pathway, so there's biosynthesis, there's assembly of the monomer, and then there's polymer, and you have to secrete it from the cell. So there's a lot of places you can interfere with this pathway to get more suberin per cell. And we've done it. So that's all I'm going to tell you. And so we've done it, and now we have to move those genes into crop plants. Okay, so can we get these other traits? Can we get more and deeper roots? And the answer is yes. So here's I'm going to show you an example first of a deeper root. So here's a plant that is what we call wild type plant, which means it came from the wild, and it is what it is. But anyways, this plant is making shallow roots. So you can see up there in the top of the soil is where suberin is staining the root red. And this is a cut through the pot. And so this is a shallow root that's meandering along the top of the soil. And so if we knock out one gene, or take out its function, or we put too much of that gene product into these plants, we get the same result. And here's when this is called gene editing, and you can see what happens. So we've turned that shallow root into a deep root. And so there are ways to do this, and there are multiple ways that we can do this, and we're working on them. So in order to do this, I just wanted to show you one fancy bit of equipment that we've had to buy. So we have a CT scanner of our own, and this allows us not to have to cut through pots. So we can go out and look at all the natural variation that nature has made in deep and shallow roots, and just ask what are the genes that contain those traits. And so anyway, so here's how we do it. We have this X-ray scanner, and it's a CT scanner, and it's huge. It's a huge piece of equipment, and all the plants come in a 5-gallon pot, you know, and they go riding through this machine. They take their image, and then the image has to be modified a lot in order to get something that really looks like a root. And so this requires people with many different traits. We need, you know, the big brutes who carry the 5-gallon pot, and then we have, you know, the people who are modifying the images that come off the scanner, and so they're more computational. And so they're the kind of wimpy people, I guess. Okay, so that's our operation to get deeper roots. Let me see. Okay, so can we get more roots? Here we go. So there are multiple ways to increase root biomass, and here's 2 ways just shown on these slides. And so here you can see on the left-hand side, the third plant from the left is a plant that's overexpressing this gene that we think is helping the plant to make bigger roots. And so it's overexpressing this gene in both the shoot and the root, but we can actually overexpress it just in the root, and we get a much more specific response. So we're probably going to do that with that gene. And on the right-hand side, you can see here's another gene that we've overexpressed in the shoot. It turns out, but it makes more root, and so that plant makes twice the amount of wet weight of root than the plant from which it came. That's almost genetically identical except for one gene. Okay, so we have multiple ways to get all of these traits. That's the bottom line. That's what we've been working on the last 2 years. We've been working hard, okay. And so where do we do our work? Well, we have a nice greenhouse up in North County that we don't really like to say exactly where it is, but I'll show you what it looks like. Oh, wait a minute. I skipped it again, okay. Here's our greenhouse. It's a 10,000 square foot greenhouse. And we're growing some of these plants there. You can see, I guess I can't really see what's growing back there, the big ones. But in this greenhouse, we're growing all the typical crop plants that are grown all around the earth. Because we need a lot of land to do this experiment. Because if you want to affect a global change, you need to have a lot of land. So there's 1.6 billion hectares of arable land on the earth. And so we need about 500 million of those hectares to be planting our ideal seeds. We have a big job ahead of us to get all those farmers on board. We'll see if we can do it. Okay, so now we're getting into the greenhouse. So we've taken these genes from this little plant, Arabidopsis, putting them into the major crop plants, you know, things like corn and wheat and rice, all the big calorie plants. And then we have to do field trials to show they're safe and everything else, right? And that we haven't lost yield. So we start that in the greenhouse, and then we go to various field sites. So here's our field sites that we currently have operating, and we have 4 of them. One's in Central Valley. We have one down in Yuma. We have some over on more toward the East Coast. One's in Kentucky, and one's in Indiana. And we'll have about 20 of these sites in the next year or 2. Okay, so we'll start to put all these plants out, and because we have to see if we're actually really pulling CO2 out of the atmosphere. And so this is going to take a few years of field trials. Okay, so, and this just shows a field experiment going on at the Yuma site, I think. Okay, so there are some co-benefits of planting these kinds of plants in the soil. So having big roots gives you increased soil carbon and plant stress resistance. You also get agriculture is not sustainable at all right now. We've had to feed so many people for so long that it's become a real drain on our agricultural lands. And so we can improve soil health by getting more carbon back into those soils. Yuma is almost completely depleted of carbon in the soil right now. They can't grow anything there but bales of hay, I think. Okay, so we can improve soil health, we can increase resistance to drought, flooding, and disease. We think we'll get consistent yield and productivity per hectare if we have these bigger, more robust roots. And it'll also improve water and nutrient use efficiency, which we need to do that because agriculture is a major contributor to greenhouse gases. Okay, all right. So we're on an aggressive timeline to meet the climate crisis challenge. Just because if we don't have something out there in 30 years removing carbon at a global scale, and it's going to take a lot of different technologies. There's a lot of talk about cutting emissions, and that's really important to do. We all need to cut our carbon footprint, but we also need, as I said at the beginning of my talk, we also need to take out the carbon that's already up there. Some of it at least. And so we have an aggressive timeline to do it. So the first 5 years we spend on research and development on gene discovery. And that's what we're in the middle of right now. We have a pretty good, powerful program going in that. But we knew we could do that. I mean, because that's what we've done for the last 30 years in the plant biology lab at Salk. But it's the next 5 years that are going to be the really challenging ones, right? Because we have to optimize all our genes so that they're giving us really good yield and stuff so we don't starving people as a result of removing CO2 from the atmosphere. But anyways, so we have to get this right. And then during that period, we have to get farmers on board to buy our seeds. Else, if they don't want to plant them, we won't be growing them. Okay, so I mean, we need 500 million acres, as I said. Or hectares. Okay, and then years 10 through 15 and beyond is when we go full scale. And so we really like this program because moving CO2 from the atmosphere, it turns out to be a very expensive process, right? And so what we do is take advantage of all the agricultural infrastructure that's already out there. So, I mean, so every country protects their farmers, right? And so they have farm subsidies and things like that. And so it can be easily set up to have a carbon bank. And so if we have a carbon bank and people are charged for using too big a footprint, then they'll go and find our farmers and they'll say, we need to buy the CO2 that you fixed on your land, right? And so this is going to be the way this all works, we hope. And so that's all going to be coming into play after year 10. And so that would be about 2030. Okay, hopefully we can do that. That's the real challenge for us. Okay, so why are we doing this now? And why didn't we get involved in this before now? All right, so it's because of where the field is, right? So molecular biologists like myself have learned a lot of mechanisms about how plants grow and how they're related to each other and stuff. And so that has brought about a revolution in plant genetics. And then that brought about the revolution in genomics, which allowed us to sequence the genomes from all these plants. There's 400,000 species of flowering plants on Earth. And so, I mean, it's very diversified lineage, out of when it became multicellular plants are. And so we had to know that about the plants so we can see how they're related to each other. And this will tell us how to go about getting the genes that we need to put into the different plants. And then finally, there's precision breeding. And this is all really been very important. So you may have heard of Norman Borlaug. And Norman Borlaug is a very famous scientist. And he's known for saving 1 billion people's lives or whatever. And that's because of the Green Revolution. And he made dwarf wheat, which didn't blow over in the wind. And that was a really big thing. And he selected for that trait by doing crosses among wheat plants in Mexico. Okay, so, but the other thing he did is Norman Borlaug basically single-handedly allowed us to grow plants anywhere on Earth. So normally plants are restricted on where they can grow by latitude. But what he did was find mutants in what's called a circadian clock. So this is how we can grow a plant anywhere on Earth. And so this is great, because now we had all those seeds from all these crosses, and this allowed us now to really go ahead and tackle a problem this big. So now you might say, well, why are we tackling climate change at the Salk Institute? Aren't you guys, this place where, oops, let me get back to this, sorry. Where you do biomedical research. And yeah, we're a place that does biomedical research, but the plant group is inspired by Jonas Salk himself. And so here's a few quotes from him, and these are quotes that I think they really motivate us, right? So, hope lies in dreams and imagination, and in the courage of those who dare to make those dreams a reality. That's something we all read every day when we come in on this left-hand side, where this beautiful building sited right for the equinox, in the fall and the spring, to have the sun and moon set over it. It's just a beautiful, creative site to be on. And so it just makes you think you can do stuff, you begin to believe in yourself when you're on the site. And another famous quote of Jonas, which is very relevant for the climate change proposals that we have on the board, is our greatest responsibility is to be good ancestors. And what can I say? If you have kids, you don't want to leave this earth in the shape that it's in right now. Okay, so that inspiration and a challenge by one of our former presidents led to this vision. So here's our vision. So this is out in the desert, and on the right-hand side we have windmills, and this is a way of making electricity that doesn't leave a big carbon footprint. And so that's a good thing, so that will cut emissions. And then we have, right beside it, we have sorghum. Sorghum is a relative of corn, and it's a great plant to grow because it doesn't need a lot of nutrients in. And so this is going to allow us, we hope, to take some of this bad land that hasn't really been kept up, and San Diego has a lot of it around us, and grow plants like sorghum so we can actually, it's used as feed for most of the world, though Africa uses it as food also. But basically sorghum is just out there growing on marginal land, so we want to develop a variety of sorghum that would be ideal and allow us to fix a lot of carbon and bury it back in the soil. Okay, so we have a good core team, and this core team is driven by mechanism. And here we are. And we're all faculty members at the Salk, and you can see we have a lot of people named Joe on our team. We have Joe Noel, Joe Ecker, and myself, Joe Chory, sometimes I'm called that. And then we have Julie Law, Wolfgang Bush, and Todd Michael, who round out the root team. So, I mean, it's a great group. We all really are passionate about this project. And we hope that we can be part of the change that will save the planet for our future generations. So that's what we're doing. And so I want to thank you for coming to the webinar today. And I think that's it. So I'll take questions now, I guess.
Joanne, thank you for a magnificent insight into plants. We'll now begin the Q&A portion of the program led by Candace Cole and Joanne Chory. Candace?
Thank you, Betsy. What a wonderful program, and so informative and so upliftingly positive in terms of what the future possibilities are to change some of the problems that we are dealing with today. I have a few questions here that have come in through pre-registration and some questions that have just been asked. If I don't get to everybody's question, I apologize. We are going to try and answer some of them after the program, and there will be a frequently asked questions section that will be sent out to everybody who registered. The first question that I have for Joanne is, please tell us how you came to choose the science of plant biology as a profession, and since March is Women's History Month, did you have any particular women mentors? And share a little about them if you did.
Okay. All right, how did I pick plants? Well, it was 1983, I think. I was finishing my PhD, and I wanted to work on something. I was a microbiologist. I worked on bacteria up until that point. And so I wanted to go and like learn something about eukaryotes, you know, these higher organisms. But human biology wasn't very far advanced. And then the model systems for human are all things like Drosophila, the fruit fly, and things like that. And those fields were very competitive, way too competitive for me. I went and visited some plant labs, you know, and they didn't know anything about how plants grow or anything. So I just like, this is a field I can make a difference. And so I went in there, and it was a little naive to think that I could, you know, because when you don't have any information, you can't ask a very sophisticated question. But anyways, we tried it, you know, we asked simple questions at first, and we were able to find out a lot of pathways in plants that allow them to grow. So that's how I picked plants and stuck with it. And so women mentors, you know, both of my advisors, my postdoc advisor as well as my PhD advisor were men. And they were both good mentors to me. I don't think they had any bias against me as a woman, even though it was the 80s and people still smoking in the lab, you know. You remember what that was like? Anyway, but anyways, so let me think. I think of mentors as coming from all walks of life. You know, I don't have one mentor really. And so I view my daughter as a mentor in some ways, right? Because I come home and I tell her about stuff that bug me at work, you know, and she'll just tell me like it is, you know, you're being a baby about that or whatever, you know. And so I like to be told how it is, you know. And it's because she's my daughter and not my graduate student, she'll tell me stuff, you know. And so she's a mentor in a way. And I had a postdoc who, while she was in my lab, you know, published this beautiful paper, solved a big problem, and had 2 kids, you know. And, you know, she was lucky that she had no complications with her pregnancy and stuff. But I really admire that because she really just focused and did her work and got a great job and left my lab, you know, on normal schedule. And so I thought she was a great role model for everybody, you know, because she just figured out how to get it done. And so I have those kinds of mentors, and they're often young women. I find young women to be very interesting. So anyway, they're not baby boomers, you know, in any sense of the word. You know, they have a whole new way of looking at the world. So.
Thank you.
Okay.
Will we be able to utilize this information in our home gardens?
I think that's the idea eventually. But I think at first we really need to get it into the, the people at Salk need to get it into the major crops because we need a lot of land to really get billions of tons of CO2 out of the atmosphere, right? But I think that would be a great idea. I think we should get Home Depot on board, you know. And so, and then have them sell, you know, carbon sequestering plants, and people will feel good about it. So even though it might not take out gigatons, it'll be involved in taking out tons, right? And so.
Will these plants be able to propagate themselves, or will farmers need to buy new seeds every year?
Well, I mean, many of the crop plants that I'm talking about are hybrids, right? And so they have to buy new seeds. So all the corn that's grown with the high yield, they're all hybrids. So you have to have some farmers just do the crosses and have them ready for the seed the farmers buy. So things like rice, no, I mean, rice will grow rice and the farmer will save some of their seed and just grow it again. It's not going to be something they have to buy every year, necessarily.
Okay. The next question that several people have asked this in various forms, since these wonderful improvements in the plants are caused by genetic engineering, is this going to be a difficulty in getting the farmers and the public to accept it, and how could this be addressed?
Yeah, this is always a big issue for us because, you know, GMOs really bother people. And so And some of the crops, they don't even allow GMO, so there are no GMO wheat. Okay, so that whole community has decided not to make genetically modified wheat. So in that case, we have to do it by some other method. And the way we're doing it is by bringing this natural variation from wild strains of wheat into the domesticated strains, right? And so you do crosses like Norman Borlaug did, you know, and you try to get the genes for these traits that I was talking about, bigger roots. We have a bigger root gene already from wheat that came out of a cross. And so we'll do it that way. And gene editing is not considered a GMO, so if you go in and modify a gene in the context of its own genome, you know, the FDA and the EPA and the USDA have all said it's fine, it's not a GMO. But so in the United States, 96% of all corn grown is already GMO. And I think soybean is 91% GMO. So those two crops are among our 6 crops that we want to grow. And so we'll probably do GMO in the United States, unless the rest of the world is still objecting so much that they won't accept carbon dioxide being removed from the atmosphere from a GMO plant. But they're already GMO, so we figure we will probably just continue to do that with those 2 crops.
Okay, so this question's been asked in several forms too. The increased root mass and the increased suberin in the roots will stay in the soil. That's the idea of keeping the carbon in the soil.
Yeah.
One question in this area is how long will that actually last in the soil before it degrades? Eventually it will degrade. And the other question is, does this impact the soil for planting future crops, or if you till the soil, does it then degrade faster? You know, in general, just please address the whole issue about the roots in the soil.
Right. So you can't till the soil. We'll just say that. So you leave the root down there in the soil, and so it's going to depend on the soil chemistry. So it's a very complicated equation, right? How much carbon and how long it will last. And permanence in soil is a very hard thing to measure, right? Because carbon passes through, you know. It doesn't all just sit there. It rains and then, you know, goes downstream. So how do you actually show that you really have increased the permanence of carbon in that soil? And so that's something we're struggling with right now. We have a lot of soil collaborators, just because none of us are experts on soils, and soil is very important in this equation. And so, let me see what else I can say about this. So some people think it's a lower quality of carbon sequestered if it's not permanent. You know, so Bill Gates is the person who's written about this. And so we get worried about that, right? Because all the nature-based solutions are not permanent. And so, all right, so how do we get around that? Well, we view ourselves more as not like the permanent end of carbon usage, right? We want to just bridge a gap between getting emissions down to net equals 0 and where we are right now. So if we have permanence in the soil of a few hundred years, when sugars are all being turned over in a couple of years, I think we'll be fine, you know what I mean? And so, and then that will be good. And so we know suberin is long-lived in soils like peat. And peat is where most of the carbon in soils is stored. So, soils have a tremendous capacity as sinks for carbon. I don't know how much you guys ever think about this, but you need soil or you can't have people, right? I mean, soil and people go hand in hand. And so, I think we have to really think about it that way. And so we have this tremendous sink. We have to get it into I don't think humus is going to be a great place to store carbon necessarily, because I think you need some carbon in the soil in order to store more in a reasonable rate, right? And so it's going to be the richer soils that we're going to store the carbon. But yeah, that's definitely, those are very good questions. And and there's all these microbes that live in the soil that contribute to the soil carbon too, as you guys probably know. And so anyways, we have to take all that into account as well. So we have a big challenge there to show what we're saying is it really going to happen. Because right now it's all just a thought experiment, right? And so, anyway, we'll see.
It's more than a thought experiment. You've done lots of work for proof of concept in this. As kind of a corollary to that, we all hear a lot about the tundra melting.
Yeah.
And there's lots of carbon that's stored in the tundra, and there's a lot of methane that could be released that way. Is this something you've thought about at all?
We're not really working on methane. You're going to have a different solution for methane. But anyways, yeah, no, that's going to be, they're calling these things carbon bombs, right? And if those things happen, I don't know what's going to happen. I mean, there's going to be disaster. You know, SIO, Scripps Institute of Oceanography, where Keeling made all those measurements of carbon, and I think his wife is still in the Del Mar Garden Club, I'm pretty sure. And so anyway, she gives gifts, I see every now and then in her husband's name. And so anyways, their son is now carrying on that curve. But anyways, what was my point here? I'm sorry, I got distracted. What were we talking about? Remind me.
We're talking about the permafrost and the
Oh, yeah.
and all that.
So that's something to really worry about, but I don't know what we can do, because I think it's already a path that's been set in motion. So, yeah. And I think, you know, the ocean rising is going to be a very serious problem. Del Mar is going to have a really serious problem, because the north side of town is all below sea level. So I don't know. Yeah.
What is your experience with gardening? And is this stuff that you would use in your own garden?
I haven't used it in my own garden so far, what we have, but I have an herb garden at my house right now. I'm not, Melissa Gann, she's the gardener, she's my next door neighbor, she's in your garden club. Anyways, she's always out in her garden. But my daughter would say, I know the inside of plants better than the outsides. You know, so, I mean, I'm always grinding them up and looking at their DNA, right? But, but if you put a you know, canola plant in front of me, would I recognize it? Maybe not, you know, I don't know. Anyway, it's pretty bad. But yeah, I mean, I'm an urban kid, you know, so I didn't grow up around farms or anything. So, it's interesting. I would say I'm not the best gardener. I should join your club and get a few tips.
You would be welcome. You would be welcome. And Melissa is certainly a stalwart of our group. This is a plant question. I have no idea if this is in your purview or not. Somebody who'd be interested in knowing if any of our native plants are especially beneficial for fighting climate change.
Yeah, I think we don't know the answer to that. I think some of those would be pretty resilient plants. They would be good for fighting climate change, I think. And so, yeah. But I don't know which ones exactly. Yeah.
Oh, yeah. Certainly one of the important things, I mean, what you're doing is, the carbon offsetting is extremely important, but equally important is improving and increasing food production. And, you know, those two goals, how do you see those fitting together?
Well, I think they fit exactly together, because that's why, I mean, the crops are actually going to be a really good source of carbon sequestration, I think. And so it's something we have to be careful about, because, you know, we don't really know what the negatives are going to be. But at least with plants, you know, if something negative starts happening, you just quit growing them, you know, or you pull them out of the ground and just burn them or whatever. You know, I don't think we're going to create a major global crisis, you know, by planting more corn that has more suberin in it. So anyway, so that's another reason why we like our initiative. You know, some people are talking about modifying the atmosphere with sulfuric acid or whatever, you know, and so if you do that, I mean, you can't go back. And so, yeah.
This is somebody else who's asking a planty question.
Okay.
Can you use this technique to increase the amount of pollen on a plant to improve pollinators' lives?
Yeah, I think you can use the techniques of breeding and, you know, and trait analysis like what we're doing for different traits. I think if you wanted to increase the ability of a pollinator to find its plant and stuff, I think you'd be altering different traits, right? You'd be altering traits in the flower. But yeah, you could do that. I mean, nature's been pretty good at it. Look at Darwin's finches, you know. I mean, yeah.
You might have addressed this specifically, and I might not have quite caught it. Do you have any idea if we were able to get the farmers on board with this, what amount of carbon would be able to be sequestered with this kind of a technique that you're...
Yeah, I'm glad you asked that because I took that slide out, but I should have said the answer to it without showing how we get to the number. But anyways, we think we can take out 4 to 8 gigatons per year. So, yeah. If we get up to 500 million hectares. Okay. So we'll see.
Are some crops more amenable to this kind of modification than others?
Yeah, definitely. So some crops are really easy to transform and make it to a GMO, right? But some of them are much more difficult, and very few people can actually do the experiment. And so that becomes an issue when you have to use some of them because, you know, we're using some of them that are difficult to actually do the transformation. So I have this one guy who worked for me for the last 30 years, and he has the golden hands, you know. And he can regenerate canola and make a transgenic canola when very few people on earth can do that. And so, I mean, because when you make a transgenic plant like that, you have to go through callus, right? So you go through callus, then you have to regenerate a plantlet. And so that's why it sometimes takes a really long time. And so, but something like Arabidopsis, that little model plant that we use to do all these studies, that is totally easy to transform. So you don't have to even make callus, you know, you just transform in the flower directly, and it gets inherited in the progeny. So
your test farms, I don't know if you call them farms, your test plots of these plants, is this in collaboration with other universities or institutes?
Yeah, so the Yuma site is a University of Arizona site. We're using a Purdue site in Indiana. We're using a University of Kentucky site. And the one in the Central Valley is a commercial site. But that one also is, again, I mean, the commercial site in some ways is better than the university sites, you know, because they have everything set up and ready to go. University sites tend to be a little more, you know, a little more easygoing, as whoever was there last feels as good as what they left it, you know. And so, yeah, so we have to be careful about how we choose the sites too. So we have a couple of soil experts who go in there and they're taking measurements now before we actually plant any of these plants. Because we're just making those crop plants that we want to put out in the fields now. So they'll be ready next year. Some of them will be ready next year to put into the field trials.
Fantastic.
Yeah.
Absolutely fantastic. So here's a question. Have you partnered with anyone in the movement for eating a more plant-based diet?
No, we haven't taken a stand on that kind of stuff.
Okay.
But I try to. I mean, I don't know. But I still eat meat sometimes, I have to admit. Okay.
I know what you mean. There are so many good questions here. We have one final question that I'm going to ask at this point, and I do appreciate everybody for asking all these great questions. And that is, as a closing question, what are the next steps that you see in your work in making this come to fruition? Pun not intended.
Okay. Okay. I think it's what I've been saying, you know, we have to get it out there on a lot of land. That's going to be the real challenge. And, you know, and we're trying to talk to the change makers out there, you know. And so we have some funding from Jeff Bezos So I called him, I asked him if I could call him recently, you know, because I think Amazon is a great example of globalizing, right? And so I asked him, you know, how do we do this? And he said, be nice to everybody because you don't know who you're going to need in the end, you know. And so when you're early, you have to just be nice to everybody. So I guess that's what he did. But that's good advice, I guess. So we take that kind of advice, you know, and we also have other people who are kind of the thought makers, you know, in these fields. And we try talking to them as much as we can because, you know, none of it, we're all scientists. We're a bunch of nerds, you know. We don't sit around in the lab all day thinking everybody thinks like a scientist, you know, but people don't all think like scientists. So you have to get out every now and then, and this is causing us to get out. So that's good. It's totally different than running my lab, though, I'll tell you that. You know, so, I mean, the way I ran my lab the last 30 years is totally different than this project. So it's been a real challenge. I've never been this busy my whole career, you know, just to keep everything straight. Anyway.
Well, best wishes with it all going forward, and thank you so much for a fascinating presentation. It's been just a wonderful hour to spend with you. I wish we could spend a couple more. Wish we could have a tour of your lab, but nobody's touring anything. There are many questions, and we don't have time to answer any more of them. In the follow-up email, Salk will be sending you a list of frequently asked questions and some other information. And we'll do our best to answer any questions that you have today that were not addressed in the presentation. Thanks for being such an attentive and enthusiastic audience. And I will now turn the microphone back to Betsy for a few closing remarks.
Thank you, Candace, and thank you, Joanne. That was a wonderful presentation, and that hour just flew by. As many of you know, philanthropy is the catalyst for new discoveries, new treatments, and new drugs. We hope that what you heard today will inspire you to become engaged at Salk in the research, as well as inspire you to create your own gardens. As Joanne has shared, the important science is continuing, and we so look forward to seeing many of you back at our campus when we can open our doors again to the community as well as tours. In conclusion, I'd like to thank our generous friends and community who support both organizations. A special thank you to the Del Mar Garden Club for providing today's platform. Special thank you to my colleague Sabrina Patterson for helping us gather all of this information. Again, Joanne, thank you so much. In the next week, we'll be sending you an email that will contain this program that you can forward to your friends, along with our contact information, and we would really appreciate getting your feedback. This concludes our program for today, and we thank you so much for attending.